Good morning, everyone. It's really great you all came in here today. We'll wait a few minutes to let a few other people connect. So let's give us to just past 8:30 before we start for today. So while we're waiting for a few more people to join in, I just wanted to welcome the Forestry Field Techniques course. Really great that you can join us today, and we look forward to having you out in the field. So this morning, we're going to go through and have a presentation from Dr. Chang. And then after that, when you come out, we'll work and look at the plot that we have out here and sort of some of the research that we've got going on as well. So hopefully you have a good day, both in terms of getting some deeper ecological knowledge and then working what we're doing out here.
Okay, good morning, everyone. So it's been, yesterday was an exciting day meeting you all and also to look at this facility. It's a wonderful place. I'm envious of your opportunity to work here. So this morning, I want to tell you a bit about my research, which you'll find very different from what you heard from Megan yesterday, and probably some of the work that is being done here. But I hope to sort of spark some interest in you to look at things a little more differently, from now on when you're working in the field. But I would really like this talk to be interactive. And so there's a chat function in Zoom. And so I would love for you to put down your thoughts in the chat session. And so that we can have and I'll try to keep that open. I don't know if I can always see your question or your comments, but I would love for you to add a comment there. So to start with, that's all try to do that. Okay, so my first question, instead of me telling you what I'm doing, is I'm gonna ask you a question. Okay, so what do you like about flowers? And especially you need to put in why you like the flowers. What's the aspect of what flowers you like and what's the aspect of flowers that you like? So go ahead and use the chat and tell us about your favorite flower and what you like about it. I'm not seeing any yet. Are you all typing? Oh, you don't have to pick a favorite. Megan has too many to pick from, so just put down an aspect, something that draws you. Okay, so we have big leaf magnolia flowers are pretty awesome. What do you like about it? What do you like about the flowers? It's huge, it's white, it's fragrant. Aristolochia is the really, well, okay, Bill said that it might not be Aristolochia anymore. So it's got a different name, hibiscus, because they taste so good in tea. Yes, I love that. Yes, it's got this acidic taste to it. Okay, great. Really fragrant flowers. So we have the taste, we have fragrance, we have, I would think, the shape of Aristolochia. And so keep it coming, okay? And so I'm gonna switch over to to share my screen now. So when I ask people, do you like flowers? I would say that I've never heard anybody who hate flowers, okay? And so we like flowers for different reasons. And so do pollinators, and so do a lot of different effects why we see such diversity in flowers. So let me share my screen. Oh, I'm not a host yet.
Yeah, Beth,
can you make me a host?
You'll have to do it. Let's see. All right.
All right, let's look at the lead plant because inflorescence look like Cheetos. All right, and when they shed pollen. That is cool. Riley says he likes lead plant. No pollination or trillium.
You're official now.
All right, I'm official now. Thank you. Okay, so keep reading the interesting posts that we have there while I share my screen. Oh. Okay. Sorry, technology challenged. All right, can you all see my screen now in a full screen format? Yes? Okay, great. Okay, so the title, I changed my title a little bit from what was announced. And so this was a title that I used for a Science Cafe talk, and I think it fits the purpose, fits my purpose here today. It's flowers more than meets the eye. And so I'm going to take you through somewhat of my own journey of studying flowers and floral variation and understanding how important it is for plant reproduction. And because of that importance, it actually is really critical, one of the critical things about understanding evolution of plants and also evolutionary ecology of different species. And so my name is Shu Mei, and I met many of you yesterday. And so this is a picture. So I took all of these pictures in one field trip to Southern Appalachian in 2019 when we were doing field work, when I was doing field work with my graduate student. And as you can see, this is just beautiful. This is one short 1 week trip. You see all these different diversity of flowering plants in the Southern Appalachian mountain region. And so we love this diversity because not only is pleasing to us, but it serves an important purpose and function in community and in the natural world. So why is diversity both at the species level and the genetic level? When I say genetic level, what I meant is by within a species level is important. It's something that we've known for some time now. There's a portfolio effect for diversity. And this is similar to, when you're investing money, you want to have a diverse portfolio, because if something is not doing well in its function, and in an investment sense, if something is not making money for you, some other similar things will be making money for you. So if you can think about in a population, in a community, when it is diverse, you have this portfolio effect that you can keep the community more resilient to changes, and it can be a evolutionarily very important function. Okay, and so in addition, genetic diversity and species diversity would also enhance or allow the evolutionary potential for species, and and also adapt to these changes that we know we are facing, as you heard from Megan's talk yesterday. So now, how is diversity and variation maintained? This is what I am most interested in, particularly in a within species, either within population or across different population level. There are multiple ways that have been proposed that variation can be maintained. It can be a non-selective process. It could be just the dispersal, they just got there and they were able to establish in a particular area and has nothing to do with selection, perhaps. There are also other explanations that are involving natural selection. And this would involve, some of the examples include the habitat heterogeneity. And this could be a spatially distributed heterogeneity, or temporally. You can have these temporal shifts of habitat or niche, as we can say. Or sometimes when you see variation in one character, it could be that this character is correlated with another character, and selection is acting on a suite of characters together, and that can maintain variation as well. And finally, there could also be balancing selection. And by balancing selection, what I meant is that you could have a distribution, different individuals with different traits, different measurement of the traits. Some is beneficial in one condition and some is beneficial in another condition. And these balancing of the sort of counterbalancing selection can maintain variation within a population. So for today's talk, I will go through real quickly and somewhat cursory in a few of the characters that my students and I have looked at, and to share with you some of our findings to show how this floral variation is important in plant reproduction. And I hope by the end of at least the first part of my talk, you're going to see that the tug of war is happening a lot, or balancing selection is happening a lot in nature. And that can be a very important mechanism for maintaining variation. The second part of my talk, I'm going to tell you a bit about a new project that we are working on. And some of these data is really just hot off the press. I just got it like 2 days ago. And so we are not really at a point to tell you the answers yet, but I'll show you some of the patterns that we've found. And also our new we're exploring how to use citizen scientist data in our research. And I hope that will be, I think that will be a good discussion at the end. Okay, so with that, the first part, I'm going to focus on the species, Ipomea purpurea. This is a common morning glory. And what the picture I have here is actually from different species of the same genus, Ipomea. Okay, as you can see, the floral characters are really different. You have the size difference, color difference, and you know, and some of the more minute differences. A little bit more about this species. It's an it's a weeding agricultural field. Okay, and so this is what we call the natural habitat. This is a soybean field. You can find them in cornfield, or you can find them in different kinds of crop fields, but it's an introduced and it's established weeds in this country for several hundred years. Its main pollinator is bumblebees. As you can see, this bumblebee is quite messy with all the pollen grains from this species on its the abdomen side. And then finally, the seed dispersal is not a very good seed disperser. The main seed disperser is actually humans. We take the seeds from this weed from field to field, and that appears to be their main dispersal mechanism. So before I talk more about the floral characters that I study, one thing that I want to remind everyone is that most of the plant species are actually hermaphroditic. And so when we say that, what we mean is that they have the ability to reproduce through both seeds and pollen grains. And so here is just a schematic flower that I drew to indicate that there are multiple ways that a plant can gain fitness. And so when we talk about fitness, it's an important measurement that basically describes how successful genetically and evolutionarily for a particular plant, okay? And so they can gain fitness through their seeds, okay? So this is what we often refer to as female function, or they can gain fitness through male function. And male function has two different types that I want to differentiate. First is that they can gain function through self-pollination, and this is indicated by the number 2 pathway here, or they can gain fitness through outcrossing, okay? And so this is, in this case, the pollen grains are being sent to another individual and produce outcross seeds that way. And so selfing rate that I'm going to refer to later on is the proportion of seeds that for a plant that is a result of this number 2 pathway and self-fertilization. Outcrossing rate will be 1 minus selfing rate. Okay, and so this basic concept has been discussed for a long, long time, since Darwin's time. Okay, and so one thing that we know, and a lot of people look for it and almost always find it, is that selfing is not so good, okay? Selfing can lead to inbreeding depression because every organism carries some genetic load. In other words, there are some deleterious genetic components in their genome. When an individual selves, that deleterious effect is exposed. And so we would see inbreeding depression very often when an individual or a population is selfing to a high degree. And so this is a very commonly and well-known sort of the scenario for selfing rate. So in the figure at the bottom here, if you have high selfing rate for a species, this species tend to have really low genetic diversity. Genetic drift will be operating at a higher importance than natural selection. Okay, and so this, because of these ill effect of selfing, high selfing, it is predicted that theoretically that species, whoops, oh, this is looking odd. Sorry, species would actually a lot of species should be highly outcrossing, okay? And so this is a figure to show the frequency distribution of different species. I'm sorry, the y-axis is cut off here, but there's a number of species that fall within a range of outcrossing rate here. And so you can see that species are either really highly selfing when inbreeding depression is not very severe, versus species would it be expected to be highly outcrossing when inbreeding depression is very important. So inbreeding depression has historically been the factor that we think is important to maintain this selfing rate variation. But since 1940s, we started to realize that there are actually a high percentage of species that fall within this, what we call the intermediate selfing rate or mixed mating system. And morning glory that I study falls within has about 70% or 30% of selfing rate and 70% of outcrossing rate. So the main question here is why? Why do we see variation in mating system, which is a very important evolutionary character, when theoretically we predict it to be one way or another, selfing or high outcrossing. And so what we propose is that even though inbreeding depression is really important and is driving, it's selecting against selfing, there could be other counteracting mechanisms that favor selfing. And so the model system that we use is this tall morning glories. And as some of you have put in the in the chat, that the first thing you see is the beautiful flowers, large flowers, the size of it, the color of it. This is all the genetic variation that you can see. In fact, I picked these flowers. They didn't all grow together, but I picked them from the field and put them together. And this is what you see when you go to a population. And so the color really attracts our attention. But it turns out that it's when you look really in detail, okay, so there's a character called anthostigma distance, and which is basically a distance between, for these flowers, these are the morning glory flowers that I cut open. There's a distance between the anthers, which produce the male gamete, and then the female surface, the stigma. And it's this distance, it's the most important character across different species when we come to estimate selfing rate. Okay, and so all these other characters, they could influence a little bit, but this is actually across different species to be the most important character. So we use this character as a surrogate of in understanding how selfing rate is maintained in terms of its variation. So we found that yes, indeed, there's inbreeding depression, but inbreeding depression by itself cannot explain what we see here. So this is a study that we looked at herbicide resistance. So this is weedy species. It turns out that this species in different population in its natural environment, that the farmers have been using glyphosate and Roundup to spray for the weeds. Some of the populations have started to develop herbicide resistance. And so when we look at herbicide resistance across these different populations that we sampled, the level of herbicide resistance did not seem to correlate with anything. Okay, so it's not correlated with the spraying frequency. It's not correlated with a lot of these abiotic factors that we looked at. But interestingly, it is correlated with the anther stigma distance. And so in this figure, what I have here is to show that when we measure the mean anther stigma distance, it correlates with the outcrossing rate within the population, as I indicated before. But when we look at the resistance to glyphosate or to Roundup level, the higher outcrossing population, so these are the dots on the top here, they tend to have lower resistance. Lower resistance versus the populations that have low outcrossing rate, they actually have high resistance. And so our conclusion from this one study is that even though selfing can suffer a lot of these inbreeding depression or the loss of genetic diversity, but if the genetic, the sort of the narrow genetic diversity that is resulted from that selection is actually beneficial. In this case, it's a human imposed strong selection on their response or their resistance to this herbicide, then selfing could be a really beneficial trait, okay? And so this goes to say that in this kind of characters, it's not a simple one way, one answer or another. And in fact, it's under different kinds of regime of selection. It could be natural selection, it could be human imposed selection that you can have, you would have these tug of war that are pulling the trait one way in some condition and another way in some condition. And this balancing effects actually allows the variation to be maintained in natural population. Okay, so that was the first trait. I should keep track of my time. So the next trait I'm going to talk about is pollen. And I know that some of you may be suffering allergy very much, like Megan has been. And so if you see this picture, you would start sneezing if you feel like you want to sneeze. But pollen is actually, you know, in addition to being annoying to some people, pollen is actually a very important character in flowering plants. And I know that pine is not a flowering plant, but I just use this to show you the visual of how terrible it could be. But pollen grain is, it carries the male gamete. Okay, so it's a gametophyte. It has sperms inside. And so, and pollen has not been has not received as much attention as it really should have been, because pollen carries half of the fitness that we were talking about before. So every seed has an egg parent and a pollen parent. So often when we look at fitness, we look at plant fitness, we look at how many seeds that plant produces, but we kind of ignore what pollen is doing, okay? And so that's an area that we know a lot less in terms of its evolution and in terms of its effect, its influence on plant fitness and plant reproduction. And so that's what I wanted to do for this second part, or what we have done for this second part. So morning glory is pollinated predominantly by bumblebees. And as you can see, they have really big pollen grains in comparison to other pollen grains. And so this bumblebee actually enters the flower, comes out all dusted by these pollen grains. But the cool thing about the system is that the pollen grains are so big, they're about 100 micron in diameter. And that is, you can think of a 1 millimeter is a 1/10 of a millimeter. So you can actually see with naked eye when they fall, when they are deposited on the petal. And so when we look at a flower, we know whether the bees have come or not, because bees are messy, they just leave a mess behind, as you can see on the flower, on the picture on the right. When they leave a mess, but they also carry a bunch of pollen grains with them, and it serves the pollinator's purpose. So this is, so a little bit more about morning glory pollen. And this is a picture that everybody uses when we want to talk about pollen grains, because it's from Wikipedia and we are all welcome to use it. And so the largest pollen grains here is actually morning glory pollen grains. Okay, and so you can see, I just labeled the other pollen grains, and you can see that they come in different shapes, different sizes. And we know that, you know, the size range is large, but we really don't know the function of a lot of the characters in pollen grains. Yes, in pollen grains. And so what I have here, the pictures on the right are are some of the high magnification pictures that we took with the morning glory pollen grains on the morning glory stigma. Okay, and you can see that you can actually see pollen tubes coming out of pollen grains. And so this is another sort of biology of pollen that I want to share with you, and that is pollen carries the male gamete inside, but it's the pollen tube that germinate from the pollen grains that have to go through the styler tissue, the female structure, to get to the egg. Okay, and so being able to germinate well, grow fast, can give advantage to pollen grains. And so in trying to study or trying to understand the male function or male fitness, yes, I said all of these, except that pollen grains are really cool because 70 to 90% of all the plant's genes are expressed in these little pollen grains. Okay, and so we often ignore them and looking at how big a plant is, how big their leaves are. But in fact, 70% to 90% of genes are expressed in pollen. And there's definitely potential for us to use that as a way to select for crop species, for example. And so going back to the study that we did is that we found in morning glory that there is a pollen size variation. Okay, and so this variation exists both within population and across different population, and it's a genetically based character. Okay, and so why is genetically based so important? Because it provides the material for evolution or for selection to act on. Okay, so given the, like what we talked about before, the genetic variation, genetic diversity is really important, and so with this genetically based trait variation, we want to know whether selection, what kind of selection agent or what kind of selection pattern might there be on this character. We also know that if you go to natural population, you look at a flower, a morning glory flower, you can find more than 100 pollen grains on there. So what this tells us is that, you know, these more than 100 grains, there are only 6 eggs or 6 ovules available in one flower. So there's a lot of competition among the pollen grains to get to the eggs. And so we set out to test why does pollen size matter? The first hypothesis we tested was that the size can actually give advantage, the advantage to larger pollen grains, because we hypothesized that, you know, the fact that they are larger, there's more resources in the pollen grains and they can grow faster. Okay, so we used the genetic lines that differ in pollen size, and then we did a pollen competition experiment in a greenhouse. And so these are our morning glory forest that we have to maintain in a greenhouse. And so, so the idea here, the setup and the idea is that my graduate student, Brittany Foltz, would put equal amount of pollen grains that are large or small on the same stigma, and as I indicated here. And there are 6 eggs here. And we put them on the stigma at the same time. And basically the idea is like running the racetrack. Okay, everybody's set and get going. Okay, and so we then evaluate the seeds to see which pollen parent actually was a successful pollen parent using genetic markers. So we have different genetic markers that we know the large pollen parent carries and the small pollen parent carried. And so we then genotype the progeny, and we found that large pollen grains in this case have it has more than 3 times as high of a reproductive success than small pollen grain. And this tells us a very clear selective advantage for large pollen grains. They run faster. Okay, so we know that this is due to their growth rate. They grow, morning glory is actually one of the fastest growing pollen tube in the flowering plant. And so, so the larger the pollen grains, the faster that these grains will grow. And this advantage then led us to ask, so why don't all plants have large pollen grains, right? Because if they're so successful, what's there to keep the small pollen grains? And we know that this variation exists in nature. And so our hypothesis was that because the pollination process is actually multiple stages, right? And so you have the pollinator has to come, carry the pollen grains away, and then they brought the pollen grains to the same stigma. So we were wondering whether during pollen transport process that the size has any differences at all. So my graduate student, Kyle Stewart, set up this experiment. And so we have our genetically different lines grown in this cage that we built in the greenhouse. And so as you can see that this is all surrounded by bumblebee proof netting. And so we have the plants in there. We have, we order a couple hives of bumblebees. You can actually do that. Keep them in the greenhouse. We then allow them to forage or visit our flowers. And now you can really see the individual pollen grains on the petal. We took the petal, we took the anthers, we took the petal, we took actually the bees, and to really carefully dissect where do the pollen grains go after they leave the flower, okay? And so we then took these samples of pollen, look at their size to assign them to either large or small pollen. We used two different kinds of plants in there. And so the result from that, and this is a really sort of artistic presentation of the experiment, that we have potted plants, we allow the bees to come, we look at how many pollen grains are left, how many pollen grains got taken away, how many of them fall off that we never were able to account for, how many of them are on the petal or to another stigma. And so the bottom line is this, okay? So what we found is that most of the pollen grains actually are stuck on the bee, okay? They never got to the next flower, or they're gone. They're dropped somewhere on the ground that we couldn't find. A small portion of them are not removed, okay? So not all pollen grains got removed. So really, and an even smaller portion of they are dropped on the corolla. So basically they came out of the anther, fall on the corolla right away. Only about 4 to 6% of the pollen grains actually made it to a stigma at all. And it can be a self stigma, like what we said before, or other stigma, okay? And so, but when we compare the small pollen and large pollen, it turns out that small pollen grains are better at hitchhiking from the flower to the target stigma, okay? And so this, even though the percentage here is really small, but it's actually 50%. If you look at the 3% versus 2%, it's actually 50% higher in small pollen grain. And so our conclusion here is, once again, is this tug of war, okay, in the pollination process. At different stage of the process, you actually have selection favoring one versus the other side of the distribution of the trait. And so the take home from this first part is that mating system or selfing rate is actually a complex trait. We often think about, we know mating system is important, but mating system can be influenced by so many different traits. By focusing on the traits and understanding the variation of the traits, we can get to understand this sort of complex dynamic of selfing rate in nature. And the second point is that small floral traits can make a big impact, okay? And so here's my call to everyone. When you see a flower from now on, hopefully you're gonna go look at the anthers and stigma distance, and you're gonna look at, the pollen, okay? Can you see anything about the pollen, any characteristic? So it's these small, minute characters that we often neglect or ignore that can actually have a big impact on plant reproduction. Nature is full of tug of wars, okay? And so that seems to be a common trend that we see in a lot of these characters. And there can be implication for agricultural breeding. And like I said, the pollen grains express 70 to 90% of the genes that we know. And we often try to select for large yield or, these characters that we want. And it takes a long time. It takes a lot of effort to do it. And so we are, one of the research effort now is to look at correlation between characters that we're interested in and then character in the pollen grains, hoping that perhaps we can use a simpler approach to select for new crops. Okay, so that's the first part. I'm not done yet. That's the first part of what I want to share with you. And I'm actually now going to go to look at the chat. Do you have any question? I'm going to pause for a bit. If you have any question that you want to ask me, now will be the time. All right, can morning glory result from asexual reproduction? It's a weed after all. So a lot of times people, we think about asexual, we think about selfing as asexual, and that, I don't know if West, you are thinking that, but if you, I mean, you're probably not referring to that. So I don't think they can do, at least in this species, asexual reproduction is not an option for this species. They do root, so that it's a viney species, and so they do root, set down roots, but that's not their majority way. The major way of reproduction is still sexual reproduction, but sexual reproduction can be selfing or outcrossing. Yeah. How similar pollen grains look? I know. Oh yes. These. Oh, okay. So I want to share with you. So this is something that's a kind of a tangential. So pollen grains, these pollen grains are cool in the sense that it's an insect pollinated species, and so you can see these really interesting sort of a sculpture on the surface, and that helps to enhance their ability to attach to the bristles on the bees. And so you often see this wind pollinated pollen grains have really smooth surface, but insect pollinated species has these spikes, and sometimes they have the hooks to help to advance their possibility of being carried away to another individuals. Yeah. Large pollen thing. Yep. Does pollen size affect offspring fitness? Very good question. We have done that experiment, and in our study, we did not see that fitness. But the sample size is pretty small, I have to say. So I think it will be important to actually look at a natural variation, so bring in the natural variation, not just our selected lines in these large versus small. Okay, so with that, I am actually gonna continue into my second part. Okay. All right, and so this is a system that we can actually find in paint rock forest. This is geranium maculatum, also called spotted leaf geranium. I started with this system examining another character, which I won't get into, but I'm gonna give you a little background about this species. Geranium maculatum, this is a perennial herb. And so there's no good estimate of how long they live. But there's a famous quote that the perennial species never die, they just kind of regenerate. And so so we don't really know how long they live, but we know that they live pretty long time. It's an understory, or it can be an edge of the woods species, but we see them in mesic environment a lot of the time. Its distribution is on the eastern North America. You can find them into Canada, but not too much further north to the border of US and Canada. The southern range, it ends at around Georgia, where I did most of my work. It attracts pollinators using both nectar and pollen. So there are some pollinators come for the nectar, but there are also some pollen collecting pollinators as well. And the pollinators is actually a suite of the generalist pollinator. And so when we say generalist pollinator, basically it's got these flowers are pretty open, so it's accessible to many different kinds of pollinators. But what we found most is small bees. Bumblebees come visit, but not as often, but small bees in the forest are their primary pollinator. So the character that I originally started with studying this species for this species is the sex expression. And so it's a really cool system that there are some populations have both females and hermaphrodites, and And you also heard about this system from Megan's talk yesterday. And so what we are most interested in this system in my lab is to understand the sex ratio variation across different populations. Why do we see 50% of females in some population, 0% of female in other population, and everything in between? Okay, and so I won't go into this. I just don't have time to cover this one. But while I was studying that system or asking questions in that area, I came across some populations I found quite strikingly different from what I was used to. So around Athens, where I started this project, all the flowers that we found in that area look like the one on the right. We have pink, pale pink petal, and yellow pollen grains. When I collected up the Appalachian Mountain in the southern range, I started seeing phenotypes or individuals that look like the one on the left here, okay? And so it's got really dark pollen grains, much darker petals. And so quickly, I was very happy to find this variation in the elevational gradient. I was like, aha, there must be some kind of selection, right? And so I can come up with a lot of hypotheses. I can say, okay, higher elevation, there's more UV, and so darker pigmentation is protecting them from the UV. Or it's colder, so darker pigmentation can absorb more heat to sort of keep the development of progeny going. But all of these hypotheses is just imaginary, okay? And so, and I think, in science, we test hypotheses, but we want to test informed hypotheses. We want our hypothesis to be making sense and with some support. And so as I look more on this character, I started finding that there is actually, these are the two extremes, but there are actually a lot of variation, both on the petal color, as you can see from the top right here, the really pale, to this magenta darker purple. But in addition to that, the pollen grains also vary, yellow to kind of creamy color. And some of them, we call them dirty yellow because it's yellow on the purple anther tissue, and to this really dark purple pollen grains. And so we wanted to know, so the first thing that we did was to try to see if what I saw in the Southern Appalachian, the elevational gradient, because this is a widely distributed species, can we see this pattern repeat in other areas or not? Okay, so we set out to do surveys, and I think this is a good sort of conventional way of trying to find variation, is that you go out to the field, you drive really hard to many different populations, hoping to get a sense of how the variation is distributed. And so my graduate student, Rachel Perez-Udell, we took this field trip in 2019, equipped with swatches. Okay, so with what I have seen, we basically took these printed out little swatches so that we can quickly evaluate the phenotype in the field without having to do a lot of analysis or complicated analysis. And so similarly, we did that with the pollen. And so you can see that I'm gonna blow this up, and you can see that the pollen grains, we kind of put this into 6 different categories, but later on at the end, we found that it's better to collapse them a little bit into 4 categories. So we use this in our field survey, trying to cover the entire range of this species. So we set out happy. We found populations, a lot of them, is on the side of the road. So that's kind of fun. But then you ran into flat tire on a Sunday when no garage was open, and then rain comes, okay? And so that generates unhappy graduate students in this kind of conditions. And so we were successful in that field season, and this is what we found, okay? And so we are using pie charts to show the color combination or color frequency within a population. And so we were able to do the eastern range of the distribution. And these pie charts indicate the proportion of different color within a population. The size of it is the size of the population. And so you can see that, you know, these lines link to the pie chart into the location here. And so the elevational gradient that we initially found holds, okay, using this data. You can see these higher elevation population tend to have darker petal, and this is a petal color. And lower elevation, these are around Athens, we see these light petal. But this is not as satisfying because this is still just a portion of the distribution. And we really wanted to, if you recall, the distribution goes into the Midwest, Midwestern states. And so we wanted to know what's the distribution look like, okay? And so, but it's a big area, okay? And so, and then we were planning to do that in 2020 and pandemic hit. And so we started thinking, well, is there another way that we can actually get to this distribution pattern? And this is when iNaturalist comes in, okay? And so we brought in a collaborator, Andrew Woodell, who help us with some of the Python coding. And so many of you probably already know that iNaturalist, how it works. Basically, this is a website or a platform that anybody can take a picture in the field and you can submit it with the location information, but you can also, you know, blur that out. But information can be submitted with the location and other indicator that they ask for. But what's cool about this data set is that we don't really have to go to Midwest because someone had already been there many times. They took a picture, they submitted it to iNaturalist. Now, anybody can submit a picture, but in order to turn that particular photograph into a research or expert grade or research grade, is that you have to have multiple people confirm that, yes, this is that particular species. So, and here is a link of my student's iNaturalist, her account. And so you can see that the pictures can be taken in many different conditions, and that became a problem for us later on. But the advantage is that now we have more than 8,000 data points that we can work with. Okay, so we downloaded this is a little workflow that we did on using this data. We downloaded the pictures that are research grade. And so in this case, for geranium maculatum, we were able to use about 6,000 records. We then gave them, using what we already have, what we already know, we characterize our color into the hue, saturation, and value. Okay, so this is HSV values that you can actually designate. This is a better way than the red, green, blue to describe a color. We use this, what we have here, we can actually extract. Okay, so this is all what we're doing here is trying to automate, okay, the process of identifying a flower and give it a number, a color coding. And so we could extract from these photographs only the petal portion, and then use this petal portion, we can then put them into different categories. So without going into the field, we actually gain about 6,000 research grade of petal information. Unfortunately, for pollen grains, it's not something that's easy. Okay, and so we had to use human eyes, and so and also a lot of photographs were terrible. You couldn't really see the pollen color. So we have a much reduced sample size for pollen color, and so but we still have about 1,200 photographs that we can use for this data set. We plotted our these points on a map. Okay, and so this is the petal color that I showed you early on, and so you can see that this data set is consistent with what we found before, that in the Southern Appalachian region, we really have a focus of really dark color, dark petal color. But as you move up north and also west, you start seeing this really intermix of dark and light color. Okay, and so when we use a machine learning algorithm to try to figure out what bioclimate data or bioclimate variable can help us to predict where a dark petal individual would be present. We found that in the range-wide distribution, there's nothing. It seems like it's pretty random, not even the elevation down here. But if we put regional, if we just focus on that region of the data, the elevation still turns out to be an important predictor for dark petal color. UV level also comes out as an important. So this all makes sense. Okay, so higher elevation, this fits with what we observed before. Okay, so what about petal? What about pollen? Pollen is what really surprised us. Okay, and so in the Southern Appalachian region, like what I said before, I saw a lot of dark pollen or purple pollen in high elevation. In low elevation, we see mainly these lighter tan color pollen grains. But as you see, when we include all the entire range, we start seeing this really clear pattern of longitude, okay? Towards the east, we have these purple or light purple pollen grains, but the whole Midwest, northern states, they actually are all yellow, a light color. And so the random forest actually revealed that longitude, UV, precipitation, both the seasonality and the driest quarter precipitation is actually important in predicting where you can find yellow versus purple pollen. And also finally, the water, there's some soil, I think it's some soil characteristic. And so the best we could do now is the water holding ability. That also is another factor that is important for this. So we were happy with this result, this outcome. And, for a while. So this year, and this is the part that I was telling you earlier, that this is actually brand new data, that my student was able to finally made her way to a broader range. Okay, so expanding from the just Appalachian, along the Appalachian populations, she now has the northeastern population and some of the Midwest population. And so for petal color, and so what we have here is, again, pie charts, okay? And so let me remind you that each of these pie is actually a population of what we call a distinct population. And so the pattern seems to still hold that in high elevation, that you have these darker petal color, but the northern populations, you have the mixture. But the darkest petal color is actually still in the Southern Appalachian range here. Now, putting these two types of data together. So one on the right, this is the conventional survey data. On the left is iNaturalist data. And I would say that, they kind of reveal different things to me, okay, at least to me. And I think this is a discussion that I hope that we would have at the end of the talk. I feel like these two data sets actually provides different information, okay? And so the data set on the right, when you go out, you took the effort to survey population, there are actually information that you cannot find from iNaturalist data, which is more abundant, okay? So the survey data actually gives us the population level information. So we know that within a population, what's the frequency of one form versus another form. And that is actually an important piece of information about processes that are happening within a population. Versus the iNaturalist data, you can see that each of the dot is actually one photograph, okay? And so we could, and we have tried to kind of divvy up this whole entire range and try to put pie charts on there. But still, when you have the summary statistics like that, you lose the information of population. You lose some of these characteristics that may be specific to population as a whole. Okay, and so this is the petal color. Let's look at the pollen color. Now, pollen color actually surprised me when we look at the population level. It appears that this streak of the purple color kind of disappear when we look at the top, northeastern population here. You still see some of these lighter purple pollen there. And once again, I think this speaks to, granted that we couldn't survey as much, as many population as the dots over here. And so, but this still tells us that, having that information, having been into the field, been into the population, knowing the population level information is actually pretty important. But what's the value of iNaturalist? To me, I think the presence absence value is really critical. And I think this level, I want to say that, this is probably one of the first studies that look at this level of detail in the photographs. A lot of times we use iNaturalist data for presence absence, for where the boundary of the distribution might be. But what I want to argue is that with these photographs, given enough analysis, we can actually see a lot more than just presence and absence. Okay, and so, but this also provides us with a guide. How do you sample your population? How do you place your population for sampling? And I think it has tremendous amount of value in that aspect. So some of you may have noticed, and this is what we noticed early on as well. So these like thousands, 8,000, in fact, there's probably more than 8,000 dots on this this map. It seems like there's certain areas is a lot denser than others. And we knew from the beginning that that's not population size. We couldn't assume that's population size. And we suspected that has something to do with whether it's a park that is easily accessible, people can go there, and 10 people will go on a Sunday and take pictures and upload them, right? And so we did this quick kind of a visual comparison. On the right here is the US population density. Okay, the map that we put on the map. And you can see that the focus seems to be pretty consistent. Okay, so that also speaks to the nature of the iNaturalist data set, that it is highly correlated with population density. Okay, and so I think it's still a valuable data set, but we need to know what's the limitation, what's the value of that data, and use that with these limitations in mind. And that's what we are currently working on to write a paper with my student and I are writing a paper to kind of point out some of these tricky aspects of using the data for scientific research, and also providing some guideline for people who want to contribute to scientific research. When you take a picture, when you want to upload your picture to iNaturalist, what kind of things should you be aware of and pay attention to so that you can be most useful to researchers? Okay, so the take home from this part is that we clearly see geographic patterns of pollen and petal color in the species. The next clear next step is to ask what factors are responsible for these patterns, and that's what I was interested to start with. Okay, when I came down this road, and so random forest, this machine learning algorithm had suggested some possible factors: UV light, precipitation, soil water hold. And these are all the factors now that we are designing experiments to test how that may influence the pollen function or the selection of pollen color. Now, what I haven't said is, and we don't have information, we don't have a lot of information, we have some, is the biotic environment. Okay, so the pollinator interaction, pollinator composition, how they interact with yellow pollen, purple pollen, dark petal, light petal. And all of these are information, are different kinds of information that came from the distribution pattern that we hope to form hypothesis and test in the future. So now, finally, non-selective factors cannot be ruled out. Okay, so if you recall the pollen distribution, it goes from kind of southwest to northeast, and one can easily imagine that post-glacial migration pattern can perhaps generate that geographic pattern. And so we are also doing a population genetic study that we would, to infer the phylogeographic pattern of the post-glacial migration from Refugia to the more northern states. And finally, the last point I want to make here is that the use of citizen scientist data. And as I discussed that, I think there are some really important strengths and some limitation of this data set. But I want to say, I think this is actually a really good opportunity to encourage sort of citizen, everyone can contribute to projects like this, and it can be useful. And I think if we get everybody to be interested and be aware of some of these wonderful diversity, genetic variation out in nature, the more we get everybody behind us, the better the whole scientific field and can move forward. So with that, I want to thank a lot of this work I kind of gloss over, but it's actually, counting pollen grains, making these pollen competition experiments require a lot of effort. And so I have an army in the past, army of graduate students and undergrad who help us with these projects. And we got funding from the Morning Glory is mainly funded by NSF and USDA, but the funding comes from a lot of small sources. And with that, I want to end with, I think I kind of broadcast this yesterday, that at UGA, we have a integrated plant sciences graduate program that if you're interested in graduate school, in any aspect of plant biology, really, we probably have faculty that can help guide you through this process. So talk to me about it if you're interested. And that's all I have. Thank you for your attention.
Thanks very much. That was wonderful. Yeah.
Thanks. All right, let me go to the chat here. Oh, have you investigated the floral pigmentation on other products? All right, so did I plant that question on you? Because I actually have a photograph, a slide for that. Okay, so I was hoping because we know that bees or a lot of insects can see UV reflectance. And so just a very crude way to look at what these flowers, and so I have 3 flowers here from light petal to dark petal, light pollen to dark pollen, and I expose them, sort of shine the UV light on them to see the reflectance. And you can see that, even though they have veins, these veins sometimes can be pronounced, but those are not really reflective at all. But anthers themselves are kind of reflective, okay? And so what does that mean? We don't know yet, but it's certainly something that we're interested in learning more about it. Comparison in iNaturalist and survey data may show bias of photographs. Yes. Who's paying rock? Okay, is that Bill? That's Bill.
Me. So it's interesting. It's something we should correct for, and something we should encourage people take representative photos.
Yes, and in fact, so what Bill was saying is that we really, and that's one thing that we're thinking about including in this paper, because especially for the pollen data, I think it might suggest that in a population, if there's one plant that is very different, people flock to that plant,
that's
right? And so you end up having overrepresentation of the unique phenotype. And so that's another thing that we are including in saying, you don't always have to, but it's human nature. We see something unique, we want to go take a picture of that. Yeah, and I think that's something that researchers need to keep in mind as well. That's a limitation, definitely. Thank you. You didn't get picture of pollen grains from the Midwest from iNaturalist tissue. Oh yes, we did actually. We did. It's just the color is really. I'm sorry, the pattern, the background, the color is not very good. Let me go back to. Here. So in this one, you can see the Midwest here. They're all really yellow. Was that your question, Wes?
You got the data from the petal color by looking at pictures on iNaturalist. You didn't go out to the Midwest, but I was surprised that people might take pictures of pollen that you could tell the color on
Oh.
iNaturalist?
Yeah, no, and that's the reason okay, so yes and no. So some people, when they take a picture, some took it from 20 feet away. And so those we excluded them, okay? So we only chose the ones that we could actually clearly see the pollen color or the anther color and use those. And so it's a much reduced data set. So it's only 1,200 photographs that we use total versus 6,000 that we were able to do for petal color.
Okay, so some pictures were close enough to actually see the pollen
Yes.
and the pollen color. Okay, great.
Yeah. Yes. And so that's another thing that we are kind of saying, okay, look, these are all that we're going to have a list of things that these are all the things that, you need to pay attention to, or you want to take. For example, it's just a very quick example. Like when I was studying female versus hermaphrodite, and I talked to naturalists, and most people can tell me where they are, but they can't really tell me any detail in the flowers. And so I think, I want to sort of promote the idea that, there are a lot more things when you dive into the flowers, a lot more things to see and really cool. And some of these can be really important for the plant ecology and evolution. So how would you avoid duplicate photograph of the same flower? So we couldn't really, I mean, the only thing that we could is that from iNaturalist, you can download the information of the person who took it, when they took it. And so this is actually really good for phenology studies because it has the time as well. And so we could use that information to call some of the duplicated photographs. And so I think that's another limitation, I think, with iNaturalist is that, like I said, if you have a national park, or if you have a park that there are some, a patch of flowers that are blooming, you're going to have so many people go in there. And that's going to overrepresent that particular areas. And so yeah, and so we're still trying, I mean, correcting for human population, I mean, it's one thing that we thought of, but there has to be some other database that we can use in order to sort of correct for some of these human biased aspects of the data set. And I think there's a lot there to be explored, but the resource is there, and I think it's valuable, but we just have to be careful on how we use it.
Similar question.
Okay.
Oh, I
Is
was
there?
gonna ask.
Okay.
So you mentioned pollinators.
Uh-huh.
I was curious if there might be like nutritional or other compound differences between the yellow versus purple pollen that might affect pollinator visitation.
Yeah, visitation, yes. So there may be some, so Riley's question was that, is there any sort of chemical differences in the pollen grains that might actually influence how pollinators may prefer one or another? And so the answer is that we really don't know yet. And so there was another, so far, the only other study that I know of that look at this pollen variation. And interestingly, they found an opposite trend. They found the darker petal in the western part of the distribution. It's a very similar distribution. It's a campanula study. And so they found purple pollen in the western part of their distribution and the yellow pollen on the eastern, or lighter pollen, they don't have yellow, on the eastern part of distribution. And so in terms of the nutritional level, I really don't know, but I do know that the purple compound is anthocyanin. And anthocyanin is sort of, it has been found to have effects of, deterrent, it's sort of a defense compound. And so that would be something interesting to look at. But so far, we know that the pollinator composition differ in higher elevation and lower elevation. And so a lot more details need to be figured out. Yeah. So is there any other questions?
Well, I had a question that was
lost in the mix, but
Okay.
do single plants produce both large and small pollen?
Oh, so you're going back to the morning glory. So Steve asked a question whether a single plant will produce large and small pollen. And the answer is no, not really. So two levels of answer. One is that individual plant produce a range of their pollen grain size, but that range is actually surprisingly narrow. Okay, and that had led to early hypotheses that we really should not see pollen size variation, being that because it's functionally very important in fertilization. And so there should be an optimal pollen size. Okay, and so
you're suggesting that there's been disruptive selection producing two peaks?
no, the two peaks is actually, no, this is a continuous trait.
Okay.
So the two different forms is basically I use the extreme forms to understand the process there. So individual plant would produce pollen, a range of pollen distribution. But when my selection lines, I basically did artificial selection experiment to generate the lines. I can easily see the difference in their pollen size by naked eyes. Yeah. Do individual plant produce the same color flower? Yeah. Okay. So that's a good question, and that's an important question. Thing that I probably forgot to mention. This is a genetically based trait, and so what we have done is that we have collected plants from different populations, brought them to the greenhouse, put them under the same environment. Because if you look at them in their native environment, yes, they produce the same color. You know, the population has the same color from one year to the next year. But if you do that in the field, you don't really know if it is just an environmental factor that is influencing the pollen color or not. So instead, what we did was that we brought the plants to the greenhouse, exposed them to the same environment. So the purple pollen individual still is purple the next year. Yellow is still the yellow next year. And so we know that this is a stable trait, regardless of the environment they lived in. We also know that it's a genetically based trait. And so we have done crosses between yellow and purple and trying. So we are in the first generation, but we need multiple generations to be able to say the genetic basis or genetic control of this trait. Another thing we do know is that even though it looks like some of the, at least in Southern Appalachian, the petal color and the pollen color are correlated. But when we look at the entire distribution, as you could see, that they are actually not linked. And so that correlation that we see in Southern Appalachian could be the result of selection that kind of make them correlated with one another. Is it?
A philosophical question.
Okay.
The morning glory work. I think it's
Okay.
so cool that you find that the more selfing populations evolved herbicide resistance faster, and that really seems like a pretty novel trait. And I was just wondering, like, what do you think about the idea that selfing is sort of an evolutionary dead end, given that result?
Yes. So that's a very good question. And we thought about this a lot too. So what Megan was asking, I don't know if you guys could hear it, is that evolutionary speaking, selfing has always been considered to be evolutionary dead end. And it's simply because selfing leads to sort of, the very little genetic diversity and reduces the evolutionary potential. And so when the environment changes, they can be wiped out. They can be, extinction can happen. And so we don't expect, and if you look at phylogenetically, the highly selfing species are at the tip of this phylogeny, suggesting that they are not likely to diversify. Okay. And so the way that we see that pattern is that this is a human imposed very strong selection, okay, in a short period of time. Okay. And so the consequence of that pattern that we see here is, we don't really know, evolutionary speaking, to this species, what is going to happen after this. But another layer of complexity is that, like I said, the distribution of seed dispersal of this plant is human mediated. Okay, so a lot of these are in the sort of landscape of human activity. And so they seeds got brought together all the time because of that. And so I think this reflects sort of the history of what glyphosate has been used so far. And I know that, Roundup is kind of, it's on its way out. There are other, they're trying to mix different things. And so I think this could lead to these fluctuating selection temporally. Yeah, that's the best we could come up with right
The
now.
argument is that once you've evolved glyphosate, if you outcross, you lose the ability to
That's
defend
right.
yourself.
That's right.
So you want to preserve it,
That's right.
and so therefore you sell.
Yeah.
Yeah,
Only
so-
only the ones that can preserve it could lead to that. Yeah.
Yeah.
That
Yeah,
high
that's right,
resistance.
That's right.
Yeah.
Yeah, so it's just wasted genes. If the only threat you've got is
Yes.
glyphosate, then
Yes. And
it's
that's
wasted
Yeah,
genes to
nail it
worry
down.
about anything else.
yes, and that's a strong, really strong selection.
Yeah, yeah.
Yeah. Yeah,
It's
so-
interesting. Yeah, it really is. It's counterintuitive until you think about
It
it,
is.
and then suddenly, wow, yeah,
Yeah,
that's
it
what's
is.
going to happen.
Right. Right.
It's a great way, and it's what humans have been doing for years in terms of isolating populations
Mhm.
for characteristics that
That
they
they prefer.
prefer.
Mhm.
And in this case, we're isolating it for things we didn't realize we were preferring.
Right. Right. Yeah. And the distribution of this species is not just in the U.S. So it's from Central America and Mexico as well. And so over there, I don't know. Actually, we don't know the population there, what's happening there. Yeah.
So that is super cool. The morning glory stuff is just super cool.
Thank you.
It's really nice. And I'll also have to say for everybody, if they can hear me, we need to do a lot more geranium maculatum collection in Alabama,
Yes.
which has the southernmost populations
Uh-huh.
of geranium maculatum really around, and very extensive throughout the state. And we think they're weird here. They're a little funky here. So we need to be doing more collecting over in Double Springs. We need to be doing it down in the Red Hills and in the Black Belt. So
I would love.
by
Yeah.
the way, that's something we need to help Shu Mei with.
Yes, please. Yes. I think Angus told me yesterday that he's already uploaded a couple geranium maculatum pictures in the past. We looked at it. It's got purple pollen. So
Isn't that
so,
interesting?
yeah.
Yeah.
So yes, I would love to have more information from this area.
Yeah. And the trait that we're seeing in the higher parts of the Appalachians,
Mm-hmm.
they actually come from historically
Mm-hmm.
from populations farther south
Yeah.
that
Yeah.
got moved into those and just in
So-
different ways.
So
it's
my
improbable.
sort of no data, no support hypothesis is that they come out of these refugia, and the ones that move to higher elevation, there's some selection favoring, and mutation happens, and then that character kind of stuck with the rest of the population as it migrates
Right.
up north, and so I think our phylogeography data can really speak to
Right.
that. We can look at the population relationship and trying to sort of recreate the pathway that they move up north, and to see if we can actually explain this pattern.
Yeah.
But I suspect that it's probably different mechanisms operating potentially in different areas. In the Southern Appalachian, selection is probably pretty important, but maybe some of the northern states or northern populations that shift, that the balance can shift. Yeah.
Can you get the same data from like digitized herbarium specimens?
Yes, and that's actually our next project with an undergrad that is starting in the fall. So we're going to try to go to the herbarium specimen. So the color actually petal color preserved pretty well. We don't know about the pollen color just yet. And so we have to look at that. Yeah.
It'd be neat to see if you see any temporal
Mhm.
trends in that
Yes.
as well.
Definitely. Yeah. Yeah. So we thought about using iNaturalist, but that's not the sort of temporal, but it's too short a time frame. Yeah, we can't see that yet. Right.
So thank you very much. I think it'd be a good time, at least for the undergrads, to drop out of here and make their way
Okay.
out to the site. And hopefully you can catch up with them. Again, today, there'll be another group out here as well. And
Absolutely.
then some of the other professors that are still online, you might want to stay in here and ask a few more questions.
Okay, thank you so much, everyone. Have a good time.
That was good.
Dr. Stone, Dr. Chen, do you guys have any other questions that you'd like to ask?
Dr. Chang, how many, or what percentage of photos on iNaturalist of flowers might also have a pollinator on there?
Oh, yes, yes. How many percentage? Well, I can only give you very anecdotal because I looked through all the pollen when when we had to do the manually. I would say, gosh, maybe less than 10%, not very many. And I think it's because it's not easy to capture. Yeah, and so we have a very small proportion of photographs that have good pollinator, and I made a note, and so that's something that we can go back to look at to help us at least get some idea of the array of pollinators that are visiting these plants. So, she does. Okay, so if no more question, then thank you all very much for listening.
Thank you.
And if there's anyone else here from A&M that would like to come out here at some point, maybe not today, as we've still got a lot of things going on, but later in the semester, it'd be wonderful to, or the summer, it'd be wonderful to have you out here. And I know, Dr. Stone, you had some other things on this month, but hopefully next month we can bring you out and you can have a look at the site and maybe meet a few more people out here.
Groovy, thank you.
Okay, so I just log out now then.